Hi, welcome to Unsupervised Learning in Python. Unsupervised learning encompasses a variety of techniques in machine learning, from clustering to dimension reduction to matrix factorization. In this course, you'll learn the fundamentals of unsupervised learning and implement the essential algorithms using scikit-learn and scipy. You will learn how to cluster, transform, visualize, and extract insights from unlabeled datasets, and end the course by building a recommender system to recommend popular musical artists. This course consists of 4 chapters, which are clustering for dataset exploration, visualization with hierarchical clustering and t-SNE, decorrelating your data and dimension reduction, and lastly, discovering interpretable features. In this video, you'll learn about 2 unsupervised learning techniques for data visualization, hierarchical clustering and t-SNE. Hierarchical clustering merges the data samples into ever-coarser clusters, yielding a tree visualization of the resulting cluster hierarchy. t-SNE maps the data samples into 2D space so that the proximity of the samples to one another can be visualized. A huge part of your work as a data scientist will be the communication of your insights to other people. Visualizations are an excellent way to share your findings, particularly with a non-technical audience. In this chapter, you'll learn about 2 unsupervised learning techniques for visualization, t-SNE and hierarchical clustering. t-SNE, which we'll consider later, creates a 2D map of any dataset, and conveys useful information about the proximity of the samples to one another. First up, however, let's learn about hierarchical clustering. You've already seen many hierarchical clusterings in the real world. For example, living things can be organized into small narrow groups, like humans, apes, snakes and lizards, or into larger, broader groups like mammals and reptiles, or even broader groups like animals and plants. These groups are contained in one another, and form a hierarchy. Analogously, hierarchical clustering arranges samples into a hierarchy of clusters. Hierarchical clustering can organize any sort of data into a hierarchy, not just samples of plants and animals. Let's consider a new type of dataset, describing how countries scored performances at the Eurovision 2016 Song Contest. The data is arranged in a rectangular array, where the rows of the array show how many points a country gave to each song. The samples in this case are the countries. The result of applying hierarchical clustering to the Eurovision scores can be visualized as a tree-like diagram called a dendrogram and quad. This single picture reveals a great deal of information about the voting behavior of countries at the Eurovision. The dendrogram groups the countries into larger and larger clusters, and many of these clusters are immediately recognizable as containing countries that are close to one another geographically, or that have close cultural or political ties, or that belong to single language group. So hierarchical clustering can produce great visualizations. But how does it work? Hierarchical clustering proceeds in steps. In the beginning, every country is its own cluster, so there are as many clusters as there are countries. At each step, the 2 closest clusters are merged. This decreases the number of clusters, and eventually, there is only one cluster left, and it contains all the countries. This process is actually a particular type of hierarchical clustering called agglomerative clustering. There is also divisive clustering, which works the other way around. We haven't defined yet what it means for 2 clusters to be close, but we'll revisit that later on. The entire process of the hierarchical clustering is encoded in the dendrogram. At the bottom, each country is in a cluster of its own. The clustering then proceeds from the bottom up. Clusters are represented as vertical lines, and a joining of vertical lines indicates a merging of clusters. To understand better, let's zoom in and look at just one part of this dendrogram. In the beginning, there are 6 clusters, each. Later on, this new cluster is merged with the cluster containing Bulgaria. Shortly after that, the clusters containing Moldova and Russia are merged, which later is in turn merged with the cluster containing Armenia. Later still, the 2 big composite clusters are merged together. This process continues until there is only one cluster left, and it contains all the countries. We'll use functions from scipy to perform a hierarchical clustering on the array of scores. Firstly, import the linkage and dendrogram functions. Then, apply the linkage function to the sample array. It's the linkage function that performs the hierarchical clustering. Notice there is an extra method parameter. We'll cover that in the next video. Now pass the output of linkage to the dendrogram function. In the next video, you'll learn how to extract information from a hierarchical clustering. Previously, we employed hierarchical clustering to create a great visualization of the voting behavior at the Eurovision. But hierarchical clustering is not only a visualization tool. In this video, you'll learn how to extract the clusters from intermediate stages of a hierarchical clustering. The cluster labels for these intermediate clusterings can then be used in further computations, such as cross-tabulations, just like the cluster labels from k-means. An intermediate stage in the hierarchical clustering is specified by choosing a height on the dendrogram. For example, choosing a height of 15 defines a clustering in which Bulgaria, Cyprus and Greece are in one cluster, Russia and Moldova are in another, and Armenia is in a cluster on its own. But what is the meaning of the height? The y-axis of the dendrogram encodes the distance between merging clusters. For example, the distance between the cluster containing Cyprus and the cluster containing Greece was approximately 6 when they were merged into a single cluster. When this new cluster was merged with the cluster containing Bulgaria, the distance between them was 12. So the height that specifies an intermediate clustering corresponds to a distance. This specifies that the hierarchical clustering should stop merging clusters when all clusters are at least this far apart. The distance between two clusters is measured using a linkage method in quat. In our example, we used complete linkage, where the distance between two clusters is the maximum of the distances between their samples. This was specified via the method parameter. There are many other linkage methods, and different linkage methods give different hierarchical clusterings. The cluster labels for any intermediate stage of the hierarchical clustering can be extracted using the f-cluster function. Let's try it out, specifying the height of 15. After performing the hierarchical clustering of the Eurovision data, import the f-cluster function. Then pass the result of the linkage function to the f-cluster function, specifying the height as the second argument. This returns a numpy array containing the cluster labels for all the countries. To inspect cluster labels, let's use a data frame to align the labels with the country names. Firstly, import pandas, then create the data frame, and then sort by cluster label, printing the result. Do note that the SciPy cluster labels start at 1, not at 0 like they do in scikit-learn. Now, we'll learn about an unsupervised learning method for visualization called t-SNE in quat. t-SNE stands for T-Distributed Stochastic Neighbor Embedding in quat. It has a complicated name, but it serves a very simple purpose. It maps samples from their high-dimensional space into a 2- or 3-dimensional space so they can visualized. While some distortion is inevitable, t-SNE does a great job of approximately representing the distances between the samples. For this reason, t-SNE is an invaluable visual aid for understanding a dataset. To see what sorts of insights are possible with t-SNE, let's look at how it performs on the iris dataset. The iris samples are in a 4-dimensional space, where each dimension corresponds to one of the 4 iris measurements, such as petal length and petal width. Now t-SNE was given only the measurements of the iris samples. In particular, it wasn't given any information about the 3 species of iris. But if we color the species differently on the scatter plot, we see that t-SNE has kept the species separate. This scatter plot gives us a new insight. However, we learn that there are 2 iris species, versicolor and virginica, whose samples are close together in space. So it could happen that the iris dataset appears to have 2 clusters, instead of 3. This is compatible with our previous examples using k-means, where we saw that a clustering with 2 clusters also had relatively low inertia, meaning tight clusters. How spread out the samples within each cluster are, can be measured by the inertia in quat. Intuitively, inertia measures how far samples are from their centroids. You can find the precise definition in the scikit-learn documentation. We want clusters that are not spread out, so lower values of the inertia are better. The inertia of a k-means model is measured automatically when any of the fit methods are called, and is available afterwards as the inertia attribute. In fact, k-means aims to place the clusters in a way that minimizes the inertia. To start with, import t-SNE and create a t-SNE object. Apply the fit_transform method to the samples, and then make a scatter plot of the result, coloring the points using the species. There are 2 aspects that deserve special attention: the fit_transform method, and the learning rate. t-SNE only has a fit_transform method. As you might expect, the fit_transform method simultaneously fits the model and transforms the data. However, t-SNE does not have separate fit and transform methods. This means that you can't extend a t-SNE map to include new samples. Instead, you have to start over each time. The second thing to notice is the learning rate. The learning rate makes the use of t-SNE more complicated than some other techniques. You may need to try different learning rates for different datasets. It is clear, however, when you've made a bad choice, because all the samples appear bunched together in the scatter plot. Normally it's enough to try a few values between 50 and 200. t-SNE only has a fit_transform method. As you might expect, the fit_transform method simultaneously fits the model and transforms the data. However, t-SNE does not have separate fit and transform methods. This means that you can't extend a t-SNE map to include new samples. Instead, you have to start over each time. The second thing to notice is the learning rate. The learning rate makes the use of t-SNE more complicated than some other techniques. You may need to try different learning rates for different datasets. It is clear, however, when you've made a bad choice, because all the samples appear bunched together in the scatter plot. Normally it's enough to try a few values between 50 and 200. A final thing to be aware of is that the axes of a t-SNE plot do not have any interpretable meaning. In fact, they are different every time t-SNE is applied, even on the same data. For example, here are 3 t-SNE plots of the scaled Piedmont wine samples, generated using the same code. Note that while the orientation of the plot is different each time, the 3 wine varieties, represented here using colors, have the same position relative to one another. Thanks for watching. In the next video of the playlist, we'll learn about the most fundamental of dimension reduction techniques, principal component analysis, PCA and QUAT. PCA is often used before supervised learning to improve model performance and generalization. It can also be useful for unsupervised learning. Also, if you have any questions, you can ask them in the comments section and we'll be pleased to answer those for better level of understanding. Like and subscribe to stay updated about our upcoming videos about artificial intelligence. See you in the next video.